Hello, everyone. Good evening. Good evening, ma'am. Okay. 6:03. Let's wait for 5 more minutes. Let other join. Then we can initiate the session. Okay. Hope the screen is visible to everyone. Okay. Yeah. So again, good evening to all. So in today's session, we are going to discuss the repair mechanisms of the DNA. So in this particular course, last week we discussed all different kinds of DNA damaging agents that will create the breaks in the genome, and it may create a double strand break in the DNA double helix or a single strand break in the DNA double helix, and how it is repaired. Okay. So last week's discussion, we got to know that our genome is continuously exposed to several kinds of DNA damage. It could be endogenous DNA damaging agents, or it could be exogenous agents that causes the DNA damage. Okay. Anybody wants to name what we discussed? What kind of endogenous DNA damages we discussed? Endogenous, that means that is like inside the cell only. Those factors are responsible to create a break in the DNA or the genomic content. Okay. So what could be the factors? For example, replication is happening, but due to the replication error, there are certain mismatches. Okay. And if those mismatches are left unrepaired, if again replication fork passes through those mismatches, it will halt there, and that halt will cause a break at that particular junction, so that it gets repaired first, then only the genome will again resume the replication process. Okay. Certain ROS are generated. Okay. Reactive oxygen species that will react with the nucleotide bases in the DNA, and that reaction will cleave the phosphodiester bond into the bases, in between the bases, and that will create a nick, which further leads to the double strand break. Okay. Other, in the exogenous damage, we read about UV induced repair. Okay. So in UV induced repair give rise to the thymidine thymidine dimer, okay, which we also referred as CPD, okay, cyclobutane pyrimidine dimers, okay. So if these dimers are created, that will also make the replication fork to halt, and it will lead to the double strand break in the DNA. Okay. And these double strand break are the most dangerous breaks to the genome. If they are left unrepaired, it will lead to several abnormalities, deletion, insertion, several different kinds of abnormalities, which may cause the cell death as well. Okay. So these breaks needs to be repaired. So in the last lecture, we discussed base excision repair, nucleotide excision repair, and mismatch repair. Okay, these processes. Other than that, the other robust processes that we are going to discuss today, those are to repair the double strand break. Okay, DSB are double strand break. Okay, so we are going to discuss today about double strand break repair. Okay? DSBR. Okay, so double strand break repair can be done by majorly by two pathways. Okay, so one pathway is homologous recombination. Okay, so homologous recombination you all also have heard during the meiotic division, right, where with the help of recombination event, it the gene diversity occurred, the exchange of gene information occurred, so that the gene diversity come to the next generations. Okay, during the sexual reproduction. So there this particular process, which is referred as homologous recombination, takes place there as well. But there, those breaks are the programmed double strand breaks in that particular aspect. Okay, so in meiosis, same process takes place, but that breaks or those double strand breaks are programmed breaks. Okay, that are uh, intentionally created by the cell so that it undergoes a homologous recombination event to produce the crossover product to bring the genetic diversity during the sexual reproduction. But if during a normal asexual cell cycle or division, at any step, if the cell faces the double strand break, it also adapts, the homologous recombination pathway. So with the homologous recombination notion, what we are referring here is, okay, so we are referring that, suppose this is a break, this is a double stranded DNA, and here it is a break. So double strand means break in both the strands of the DNA. We all know that our DNA is a double stranded helical DNA. So if the break is in both the strands, then it is referred as double strand break. If there is a break only in one strand, okay, like out of these two strands, only one strand is facing the break, then it is called single strand break. Okay, and if it is in both the strand, it is double strand break. So single strand break we do not refer, we generally refer this only in one strand break, we refer it as nick. Okay, nick in one particular strand, that is called, that is a single strand break. Okay, so these double strand breaks, which normally during the mitotic division or the asexual division the cell faces, these DSBs are also repaired by the homologous recombination pathway. That is nothing but the cell uses another intact uh, uh, homologous means similar strand. So let me draw it here. So this is one particular chromosome. Okay, and this is another chromosome. Okay, so suppose if, so suppose if this particular one, here there is a, everything got erased. Sorry, let me make it again. Okay. And this is another chromosome. Okay. So suppose here in this one particular chromosome, it faces a break here. Okay. So then what happens? And like suppose in zoom it looks like this. Okay, it faces a break here, then it can use a similar, because it is another homologous chromosome of it. So here also the sequences of on this chromosome is identical, or you can say similar to this one. So the other strand can help, or this particular break to be repaired. Okay, or in also similarly in this particular DNA also, suppose one, if it is a, okay, this is, these are sister chromatids, so one chromatid, this is another chromatid. So then in that case also, if here it is a break, it, this particular strand use the other sister chromatid strand which is intact, use it to repair this particular break. And we are going to discuss today how this particular pathway takes place. Okay, so either you use a homologous chromosome, okay, which is a where the homologous sequence is present to repair the double strand break, or you can use a intact sister chromatid as well to repair the double strand breaks. Okay, so that another portion of the chromosome which is intact should be homologous to the broken sequence, then only the break will be repaired. So that is why this pathway is called as homologous recombination pathway. Okay, so this is one pathway. Other pathway we have, so this is homologous recombination pathway. Other pathway is non-homologous end joining, which is referred as NHEJ, non-homologous end joining. Okay, so there what happens if there is a break, okay, in the DNA. It doesn't need any homologous template or homologous chromosome or homologous DNA to repair this particular break. There are certain proteins involved, okay, which will act upon this particular break, and that will lead to joining of this broken ends. Okay, but there is a chance that when these two things or these two broken ends are joining, there are chances of deletion. For example, so like this is broken, but there are chances that before these two ends are being joined, this portion, the end portion gets deleted or degraded by the nuclease's activities. Then also if the fragment is joined, now it became smaller because some of the information which was contained here, those genetic information is now lost. So it may also lead to deletion, deletion of gene, deletion of nucleotides, which may cause mutation, or it can also be something else can also be incorporated when the break is repaired. Okay, because it is just joining of two open DNA ends. So when the joining is happening, in between something else can also be integrated. So that will lead to insertion, another type of mutation, you can say. Either it can lead to deletion or insertion. So this non-homologous end joining is more error prone compared to the homologous recombination pathway. But in higher organism, this particular NHEJ pathway is more preferred than HR. Okay. So these are the major two types. Okay. And if the cell failed to repair the HR, the break via HR or NHEJ, it opts for another pathway that is called alternative end joining. Okay. Also refers as alt EJ, or it is also referred as BEJ because it is a backup pathway if HR and non-homologous end joining both fail to function. So it is also a backup end joining or alternative end joining or microhomology mediated end joining pathway. Okay, so this is the third pathway. So let's discuss about these first. So let's begin with HR. Okay, so here, yeah, here you can see HR, NHEJ, all TJ. Okay, so here you can see in HR, you have these complexes MRN, CTIP complex, which recognizes the double strand break. Then it undergoes the resection. Then there are certain proteins which are uh, binding there that will lead to strand invasion and homology search. And with the other strand, which is the homologous strand here, with the help of that, this particular strand is now repaired because the broken part is synthesized and again it rejoins as the old one. Okay. Similarly, in NSGJ, there are different proteins which will simply ligate the broken ends and it doesn't need any sister chromatid or homologous chromosome. Okay. And then we have alt EJ, where the complete different procedure, resection, alignment of short homologous sequence, and then ligation of the broken DNA ends. Okay. So these are the basic three pathways. I hope it is clear till here for everyone. If you are having any doubt, please ask me. Yeah. I'll explain all the processes in the detail. Like, Just, I think I'm not able to hear you guys. Okay, so I think your question was to repeat those pathways, right? Like exact mechanism or something. Yes, ma'am. Yeah. No problem. Let's discuss one by one in detail. Okay. So I'll initiate with homologous recombination. So homologous recombination is a conserved pathway from bacteria to the higher eukaryotes. Okay. So the proteins are also very much conserved. Okay. So let's first discuss about prokaryotes. Okay. So how it happens. So I'll explain you in a short and then we'll go in detail with those parts. So here is a broken end. So one, there is a break. First thing is recognition of those break. Okay. Then after the recognition, it undergoes resection. Resection means resectioning means just breaking a certain portion from that double-stranded helix. Okay. Suppose it is a 5 prime to 3 prime, 5 prime. So it is something like this. Wait. Okay. So here you can see after the resectioning, that means this particular portion is being uh, fragmented or deleted or cleaved. Okay. So to generate the 3 prime overhangs. Okay. So these are one of the important steps. 3 prime overhangs are generated. Then after this overhangs are generated, then the homology search will initiate. Okay. So once the homology search is initiated, that means finding a homologous copy. So suppose this is the homologous copy. This is the homologous copy. Just a minute. Stopped working. 1 minute, please. The pen stopped working. Let me do it. this only. Okay. Yeah. So this is now the process. Okay. So once the homologous chromosome is found, then what happens? Then uh, the strand invasion takes place. That means this particular 3' overhang which is generated here, that will invade the homologous chromosome. Okay. So it will look something like this. Invade. Okay. So this is how it invades the other uh, particular chromosome. So here, certain helicases are also involved, which will resolve this particular structure. So now you can see. So this is a 3' overhang. So we know the DNA synthesis takes place from 5' to 3' direction. That means the 3' overhang is needed so that the DNA synthesis will take place. Okay, so now this particular strand which is invaded will use this particular the blue strand or the homologous strand as a template, and here another polymerase comes which will use this particular strand as a template and resynthesizes the DNA. Okay, so now you can see the because this is a homologous chromosome, it is the sequence here is similar or homologous to the sequence that has been lost during the break. Okay, so the cell uses this another strand as a template, homologous strand as a template, and resynthesizes the broken part. Okay, and that is how the break or the lost genetic information is recovered. Okay, and once the this synthesis is done, again it will be resolved like this, and similarly from the top portion also it is repaired. Okay, and then it will look, it will look like this. Okay, so that is how the broken portion is being repaired. Okay, so let's see the proteins that are involved here in this particular pathway. I hope here it is clear for everyone. What is the basic procedure? Okay. Pen is not working for this. Okay, let's see. Okay, so in prokaryotes, the proteins that are involved. So first thing is recognition and resection. Okay, so there is a complex of the protein that is firstly, so it is an mRNA complex. Okay, that recognizes the breaks. Okay, so mRNA means three proteins. Okay, so these proteins are MRE11, MRE11, RAD50, and NBS1. So this is a complex that recognizes the break. So once the break is created, if on the break mRNA complex is recruited, then the break is repaired by the HR pathway. If on the break, the break is recognized by Q complex, it is another set of protein which is Q proteins. If the break is recognized by Q proteins, then the break is repaired by non-homologous end joining. So from there is the at the recognition only the fate is decided, the broken DNA, how it is repaired. If mRNA complex is recognizing the break, it is repaired by the HR pathway. Is Q complex is recognizing the break, which is it will be repaired by the non-homologous end joining pathway. Okay. So yeah. So we have a complex here in prokaryotes, which is REC B C D complex here. Okay. So this is a REC BCD complex. What it does here is, so when the break happens, this REC BCDs are three different proteins, three REC proteins, REC B, REC C, and D. So B, C, and D. So this particular complex gets recruited at the broken end. Suppose this is a broken end here. Okay. So this trio or this complex is getting recruited. And then what it does, it has a motor activity. This particular protein has a motor activity. So this protein starts moving in this particular direction. Okay. For the resection purpose. Okay. So it is a bipolar protein. It can move in any direction. So once the break is recognized by these three proteins start moving. So the particular B protein, it is a helicase plus nuclease. Okay, helicase with nuclease. D, I guess it is only I'm not able to recall. I think it is only a nuclease. And C is a protein which is a chi-site recognition protein. So chi-site is nothing. So in prokaryotes, recombination takes place at only one particular site, which is a specific 4-8 base pair sequences, which is referred as chi-site. Okay. So once the break is created, these three proteins are recruited and start unzipping the DNA. Okay. And so as they start moving, so they're with their nuclear property. So yeah. So here you can see in this particular one. So REC B, which is an helicase as well as a nuclease. So the N-terminal portion of the protein B is a helicase and the C-terminal portion is a nuclease. Okay. REC D is only a helicase. Okay. And REC C is having a specific sequence that recognizes the chi site. Okay. So here it is written REC C recognizes chi site and REC D stops and then signal REC B to cleave DNA. REC B cleaves and continue unwinding the DNA loads REC C. So yeah. So as it starts moving, when these complexes, so it starts unzipping. Okay. Here, as it moves, this complex starts unzipping the DNA until it reaches this particular chi site. Okay. Here at the chi site it reaches. So as it unzip the DNA, it also cleaves the DNA. Okay. So if this is a 3' end and this is a 5' end, so the cleavage process or the nucleus process of this particular B and D protein is more vigorous towards this particular end. So here it is more vigorous. However, this particular end, it is not that vigorous. But both the ends gets cleaved until this trio of protein recognizes the chi site. Okay. And once the chi site is being recognized, then what happens? Then uh, only uh, yeah. So after the chi site, when the protein starts moving, so what happens? Then only this particular 5 prime overhang portion is only getting cleaved. It will not cleave further this 3 prime overhang. Okay, as it start moving from this particular chi site. Okay, and these chi sites are the particular point where the recombination takes place. That is why these chi sites are called as recombination hotspots. Okay. 1 minute, please. Hmm. So then what happens because of that particular reason, beyond this chi site, beyond this chi site, the only the 5 prime end is being resected and 3 prime overhang is being protected. Okay. And then as the DNA passes, or as the chi site reaches after a resection, after a resection, and then the now finish. Okay, sorry. Yeah, so once the chi site is passed, then these three proteins get dissociated with each other. Their function is now over. So this particular complex become non-functional. So this is how in bacteria these 3 prime overhangs are generated with the help of Rec B, C, D proteins. So Rec B has helicase plus nuclease activity. D has only uh, helicase activity when it reaches the so once the rec C recognizes the chi sequence, it signals rec D to stop, and rec D stops and then signals rec B to cleave the DNA. So rec B only cleaves the 5 prime overhang, and then uh, it cleaves and continue the unwinding DNA, and then another protein which comes on those DNA is a rec A protein. So yeah. So now with the help of those three proteins, these 3 prime overhangs are generated, and those three proteins are now dissociated from each other and unloaded from the DNA. Then to protect this particular DNA fragment from the other exonucleases, this sort of protein which is referred as Rec A protein that comes and loaded to this 3 prime overhang. So this Rec A is a highly conserved protein from lower prokaryotes to higher eukaryotes. Okay. In eukaryotes, the Rec A homologue is referred as RAD51. Okay. This is very important. Okay. So now the Rec A is being loaded, and then Rec A only performs the strand exchange, okay, or strand invasion or exchange, and homology search. Okay. So, yeah. Okay. So for the homology search, what the step is, so this Rec A protein, if I'll draw, okay. So you all know this particular portion of, if this is how I'm writing a particular amino acid from 1 to some 300, it is some approximate 350 amino acid protein. So this is the N terminal of the protein, and this particular portion is referred as the C terminal of the protein. Okay, because of the amino acid structure. You know that one side it is amino moiety, another side is carboxylic moiety, right? So this left side portion is the N terminal domain, N terminal. The end portion is C terminal of any amino acid. So this particular protein, if I have to divide, it has N terminal this region, it has C terminal region, and in between these Here it is having a DNA binding site, ATP binding site. Okay. Yeah. And one more thing. So here this particular process was ATP driven. That means in this particular resectioning process, ATP is involved. Okay. So similarly here also, this there is a site which is for ATP binding, uh, which helps in strand invasion and homology search. Then we have a site 1 on this protein and also site 2. Okay. So, as I have mentioned, after the rec BCD complex gets removed, rec A binds to this 3' overhang, which is single stranded DNA. Okay. So, it has a single stranded DNA binding site. It has double stranded DNA binding site to it. Okay. So, these are also present in this in between middle domain of this protein. So, then what happens? This once loaded on this single stranded DNA, ATP comes and bind to RECA. Okay. So here, like to all the RECA proteins, ATP molecules bind and stabilizes this protein DNA complex. Okay. ATP is bound and stabilized to this DNA and protein complex, which is referred as nucleoprotein filament. Okay, it is a DNA plus protein. It is a nucleoprotein filament. Okay, so once the ATP is bound, then what happens? Then the uh, what to say? The homology search initiates. Okay, so then the filament is getting activated, and after that, what happens? First, uh, so another chromosome. It starts searching. It search for another chromosome. So as I said, this protein has a single stranded DNA binding site as well as double stranded DNA binding site. So with the help of single stranded DNA binding site, it is bound to this ssDNA and makes a nucleoprotein filament where ATP binds as a cofactor and activates this filament and stabilize and activates these filaments. Once it gets activated, then the double strand DNA is being searched. The homologous DNA is being searched. So to the double stranded DNA, first the N-terminal portion of this protein binds. Then after that, C-terminal portion of the protein is bind. And after that, the C-terminal portion transfer the double-stranded DNA to the double-stranded DNA binding site, site 2. Okay, double-stranded DNA binding site. So, um, hmm. So if after the double-stranded DNA, the double-stranded DNA, which is bound to this RECA protein, if it is homologous, then only the strand exchange will take place. If not homologous, this DNA will be left from the RECA protein or unloaded from the RECA protein, and then it will search for another homologous chromosome. I think it is clear till now. Anyone asking anything? I'm sorry, screen is not visible. Is everyone facing the same problem? Is my screen visible to everyone? Please tell me. Is it visible? Okay. The person who asked about the screen, is it visible to you now? Is it visible? Shall I proceed? Shall I proceed ahead? Anybody give me reply? Is it visible? Okay, so I should proceed, right? Okay, so this is all about the Recce protein. So this is how the homology search is happening by binding to the double stranded DNA. And if the double stranded DNA is homologous, then only that double stranded DNA will be used as a first strand exchange. Otherwise, this double stranded DNA will be left. Okay, and other double stranded DNA search will start. So this is the Recce protein. Okay, so as we have initiated, so once the double strand break is created, then we have the REC BCD complex, which will unwind and with its with helicase activity and with near the chi site only. Okay, this 3 trio of the protein is loaded. Beyond the chi site, it will create a 3' overhang. Okay, it will create a 3' overhang like this. And on these overhangs, the Recce protein is now loaded. Okay, and homology search will begin. So if this is the homologous chromosome, again, after the homologous chromosome is after the homologous chromosome, the 3' end. Okay, the 3' end is now invaded to the, and it will create a D loop like this. Okay, so this is referred as D loop or displacement loop. Okay, because it is displacing the this particular strand from here and invading, so it is a D loop. And this particular junction here which is created is referred as holiday junction, discovered by scientist Robin Holiday. So that is why it is a holiday junction. Okay, so now this particular strand of here, which is a homologous strand, which is having a similar sequence of the opposite strand, is used as a template, and the polymerase delta will start synthesizing the DNA. Okay, and then after this particular uh, section is resolved, then you'll get the recombined products, which will be something like this. Okay, so this is what is the total HR pathway: Reg BCD, Reg A, and other polymerases, then ligases will ligate, and that's it. Okay, so now as this particular pathway is used during the meiotic division, as well as I have mentioned earlier also, so there it is used to create the genetic diversity. Okay, to exchange the genetic information. So, if during the normal asexual production or meiotic division, if there also the genetic information exchange will take place, during the asexual stages, it will create a lot of abnormalities because it will not maintain the genomic integrity. So that is why there is a check at this position. So till here we have talked. You can see D loop is created. Okay, D loop is created, the other template is being used to synthesize the broken part. And now what happens? This may lead to this pathway, which is synthesis dependent strand annealing. Okay, where the D-loop dissociation will happen. Like this D-loop will dissociated and annealed back. This particular portion is annealed back to the original broken DNA. And this portion is being amplified using this as a template. And this DNA synthesis will take place. This is a non-crossover product. But if this particular polymerization will further extend, that means this further get resolved, this D loop is resolved, and then the second end is also getting captured. This second end also gets captured here. Okay, you can see. And now this end is being used as a template here on the top. Okay, this will be create two holiday junction, which is called a double holiday junction. So double holiday junctions are formed when the crossover products are being formed. Double holiday junction. So then these holiday junctions are resolved and it will give rise to non-crossover and crossover products. Okay. So if you'll see this one as a resolved structure here, you'll see like this. If it is a horizontally, it is resolved. There is a gene. This you can see A and B, one at one chromosome, small b, small a, or another chromosome. So originally it is AB, and on another chromosome it is AB, like this. So here you can see if the holiday junctions are being resolved horizontally, the break is here, then you can see it is ligated like this, and here there is no crossover or non-recombination product is being formed. But if they are being resolved vertically, that means another chromosome's portion is being integrated here, you can see. And that is how now this you can see the allelic information is changed. So this is the recombinant product is being formed. So this is the difference. At this, after this point, it may go to the synthesis dependent strand annealing pathway where the D loop is dissociated, or it will further capture the second end and make a double holiday junction to form the either crossover or non-crossover mixed product. Okay, so this is the I would say the homologous recombination process, the entire process. Then we have the non-homologous end joining. Okay, so here, as I have mentioned, double strand break is created. Okay, then this particular double strand break is recognized by proteins which are Q70 and Q80. Okay, so Q proteins, which is a dimer of Q70 and Q80 proteins. This is in eukaryotes. Okay, so before that, let me explain you the HR pathway in the higher eukaryotes. So we read till now this RIC-BCD complex. In higher eukaryotes, the proteins are different for the HR. So once the break is created, you have the protein to recognize it is mRNA complex. Okay. That after that, it is a protein which is referred as BRCA1. Okay. BRCA1 is recruited to this broken ends, and this BRCA1, which further helps to load the helps in resectioning. Okay, so here there the it was a together rec BCD complex which was involved in the resectioning, but in eukaryotes it is a BRCA1 protein which does the function of resectioning, and it has a opposing role with other protein which is named as P53 binding protein 53 one something. This is a protein. So this protein inhibits BRCA1. If BRCA1 is inhibited, cell and follows the NHEG pathway. If not, then HR will proceed. Okay, so this BRCA1 is involved in the resectioning process again, and loading of the protein, RECA protein, which is referred as. Okay, so once the 3' overhangs are generated, there is a protein named RPA. RPA is loaded, okay, which has higher affinity to the protein. So then the RECA homolog, which is RAD51 in eukaryotes, that has the activity to perform the strand exchange and homology search. So this RPA is nothing but single stranded DNA binding protein. So as soon as a single stranded DNA is created, this RPA gets loaded to the single stranded DNA, which will be removed and displaced by RAD51 protein, which has the strand exchange activity. So then the RAD51 replaces RPA and gets loaded on the single stranded DNA. Okay, and because RPA has a higher affinity to the single stranded DNA compared to RAD51, there are other accessory protein which is referred as RAD51 paralogs are needed. RAD51 paralogs are needed to stabilize RAD51 protein on the single stranded DNA. These paralogs are RAD55, RAD57 paralogs, which has uh, the sequence similarity. You can see some conserved domains are present that are similar to RAD51, but they are not RAD51, or they do not perform the function of RAD51. They are accessory protein only to stabilize RAD51 binding to the ssDNA. RAD51, there is another protein which refers shoe complex. There are several different kinds of protein, and also BRCA2. BRCA2 is one of the most prominent protein which uh, stabilizes the RAD51 binding to the single stranded DNA. And once the binding and strand invasion takes place by RAD51, then again there are proteins like um, DNA polymerase, which synthesizes the strand, and then dissociation of D loop, and then ligase comes in the picture that will anneal those, or sorry, ligate those strands. Okay. Now NHEJ. So NHEJ is a where you don't need a homologous chromosome or homologous chromatid, sister chromatid. So there you have this heterodimer of Q70 and Q80, which binds to this broken DNA. Okay, this Q70 and Q80 further recruits another protein which is DNA PKCS. Okay, so this particular protein only cleaves this one cleaves the DNA, another protein which is this one, which is DNA PK, which cleaves off which this is nothing but is a protein kinase. Yeah, after the resectioning, this is a protein kinase. So it uh, uh, phosphorylates the downstream protein. So downstream protein is another protein which is Artemis, referred as Artemis. So this first it gets bind to the Cu70 Cu80 heterodimer, then recruits Artemis protein. So this Artemis protein is recruited here, which will be phosphorylated by the function of this protein kinase catalytic subunit. After the phosphorylation, it cleaves off the overhangs and then join these two ends with the help of XRCC and ligase 4 and then gets dissociated. So here XRCC4 and ligase 4, these are the proteins which are involved in the ligation. So this is simple non-homologous end joining, but it is prone to deletions, mutations, insertions, because now the lost sequence cannot be recovered. However, in HR pathway, you saw that the lost sequence was able to recover because the another sequence was behaving as a template to synthesize the lost DNA information. Okay. So here in the table, you can see in prokaryotes, these proteins, tool-built proteins are Cu30 and 40. In yeast and in multicellular eukaryotes, it is Cu70 and 80. Okay. And here the polymerase is Pol4, Polmo and Lambda. Nucleases are Artemis and DNA protein kinase catalytic subunit. It also has this PNK phosphatases, and the ligases are XRCC4 and DNA ligase 4 XLF. This is the ligase complex. Okay. So this is the comparison between prokaryotes and the eukaryotes. Okay. So now let's move ahead to the question and answer. Okay. So now the first question is, which one of the following is true for the primary structure of REC-BCD enzyme? Okay. So in REC-BCD, we have three enzymes, right? B, D, and C. Okay. So B, which is from a helicase, SF1 helicase family. It has helicase and nuclease domain. This is N terminal, this is C terminal. The D is only a helicase, and the C protein, REC-C, which is a protein which is for a chi-site recognition, which has sequence for chi-site recognition. Okay. So these are the three proteins of REC-BCD enzyme. So now seeing the options, let me add that. Okay. Now seeing the options, tell me which particular option is correct. First is the N-terminal domain contains nuclease motif. Is this correct? Answer. The N-terminal domain of these three proteins contains the nuclease motif. Is it correct? The C-terminal domain contains nuclease motif. Correct or not? Yeah. So the C-terminal domain contains both nuclease and helicase motif. No. The C-terminal domain contains helicase motif. No. Yeah. So this is the one which it followed, where the C-terminal domain contains the nuclease motif. I think, let me check this. D, it is only helicase. Yeah, next. In which of the following strategy a double strand break present in one chromatid is repaired using its intact sister chromatid as a template? Give me the correct answer. Yes, it is the homologous recombination. Okay. Yeah, correct. Next is the REC-C protein has 1122 amino acid residue and contains the, out of these four, which one? Nucleus motif, chi recognition site, or SF1 helicase motif, or all of the above? Yes, the chi recognition site. Good. Okay, so now which of the following is not a part of the REC-BCD enzyme complex? Which one? Yeah, correct. Other this B, D, and C are the complex, like BCD enzyme complex. Okay, and what is the function of REC-A? Can anybody tell me? Okay, what is the function of REC-BCD enzyme complex? Mm-hmm. Hmm. It is a helicase that also function as a helicase. Yeah. Yeah. So BCD first recruited to the broken DNA with the help and it unzips the broken end until it finds a chi site with the help of REC-C, and then it creates a 3' overhang where the REC-A protein is being loaded, and the REC-A performs the homology search and stand invasion. Good. Okay. So which one of the following enzyme is involved in the prokaryotic polymerase NHEJ component? Okay. Yeah, the Q30 and 40. In eukaryotes, it is Q70 and 80. Okay. Yeah. So, who proposed the eponymous holiday junction model? DNA recombination intermediate. It is Dr. Robin Holiday. Okay. Yeah. Okay. So, after all these, so now we have discussed HR and non-homologous end joining. There is one more mechanism which is referred as site-specific recombination. Okay, so site-specific recombination is less complex, present, and more specific. Okay, less complex because it involves less number of proteins. Like you saw, no, there was an array of proteins in the HR, or in NHEJ also there are certain proteins are involved. Okay, so there are RAD51, then RAD51 paralogs, then the downstream proteins, downstream helicases, which are involved in resolution or dissolution of the holiday junctions that are formed in more complex steps. So compared to that, this site specific recombination or SSR is a less complex pathway which involves less number of proteins, and also it is more specific. Okay, like there is certain sequences which are being recognized here. So this is also a recombination, but here, okay, this image is not very clear, but okay. So if there is an one circular DNA and another linear DNA is present, these sites are the recognition sites. Okay, these are the recognition sites which undergo recombination, and with the recombination this will lead to the insertion. So now with the recombination event, this particular uh, piece of DNA or the circular DNA is now integrated to this particular single stranded DNA. Okay, then vice versa can also lead to the uh, deletion. Okay, because these and this is the recombination site, and if they get aligned in such a way like this by a crossover, this particular portion can be deleted. Okay, and this will lead to the deletion phenotype, and similarly inversion. Okay, so this is one particular site-specific recombination leads to integration, deletion, inversion, all those different. Okay, so it is divided into 4 families. Okay, one is tyrosine, uh, tyrosine recombinase, and another is the serine family, serine specific and tyrosine specific. So if we'll see the basic, okay, so the overview if I'll give, for example, phage DNA. Okay, so this particular phage DNA, suppose, so in bacteriophage we know that it is a virus and it attacks the bacteria and integrate its genomic DNA or to the bacterial DNA. Okay, so that particular process is done by the site specific recombination. So there are certain specific sites which are there present in the phage DNA as well as in the bacterial DNA. So this is, you can see, so this is attachment site. ATT is attachment site, so this is the phage DNA, so it is attachment P for phage. So this is ATTP site which is present in the phage DNA, and this is ATTB site which is B means bacteria, it is present in the bacterial DNA. So these two sites are present, and that take with the help of this O region, they get part into the this crossover or recombination, and then this particular entire circular DNA gets integrated to this uh, bacterial DNA, and this is a prophage, and these sites are now called ATTL and ATTR. Okay, so so this is the basic mechanism. If I have to give the detailed mechanism, so in detail if I'll tell, so if, okay, so now there is a protein which is host specific. So this, there the differences coming with the some in this type of classification. Okay, so there are 4 classifications. So some require the host specific protein, some of them doesn't require the host specific protein. So there is a protein which is named integrase protein. So this integrase protein makes a nick here in both the strands. So how the nick is created, like which particular recombinase is creating a nick? It is tyrosine recombinase. That means in the structure of this protein, the active site is having tyrosine, and that is creating a nick. However, another is the serine recombinase. That means in the active site it is serine. So in the double strand in the DNA we discussed, so base, so phosphate with this OH, the phosphate is there, right? And another base. So this particular, any of these enzyme will attack at this position, okay, with this serine or tyrosine residue, and then it will break this phosphodiester bond. Okay, it will break this phosphodiester bond. Mhm. And it will make, okay, now there is a nick created. Okay, this is the DNA phosphodiester bond is broken, and now the nick is created here. Okay, nick means nick is created. Okay, and once the nick is created, then the crossover happens. this. It's like this, the crossover happens, and because of those crossover, these holiday junctions are dissolved and recombination will take place. So this is the basic mechanism. Okay, so the similar technique that you saw, nick is created and then the crossover will take place, the entire integration will take place. Okay, so there are several types in this. For example, this particular one required the host factors. Okay, it is host factor dependent, but there are certain systems are generated which doesn't require the host factor, where you can produce those enzymes. So there is a 3-log system and there is a FLP FRT system. Okay, so CRE, which is full form is causes recombination. So there the INT enzyme was present, which was causing the recombination. So there it is. Here it is CRE recombinase. CRE recombinase, and a flippase enzyme, which is a recombinase. Okay, so these are the recombinases, and the site that they recognize are the log site. So the CRE recombinase recognizes the LOX site. So log site is something like this. Okay, on the DNA it is a log site. So lo logs is locus of crossover. Okay, locus of crossover. So it is basically a logs P1 site which has derived from the bacteriophage phase 1. Phase 1. Okay, it is a LOX P1 site. Okay. Similarly, in FLFRT system, this particular FLFRT is FRT is the recognition site there. Okay, so similarly, LOXP site is being recognized by uh, the CRE recombinase, and there it performs the crossover, and then it may lead to deletion, and the other sequence is being deleted via the recombination. So these are the systems that are being generated to create a knockout. So these are the very specific sites that are introduced into the genome, and the CRE recombinase is overexpressed, and when the CRE recombinase is expressed, it recognizes the LOXP site, make the crossover to work, and then excise that particular genomic content that is present inside. Okay, so this is how the CRE recombinase or FLP FRT recombinase is used. Okay, so CRE recombinase is the source of this CRE recombinase is the Phage1 DNA, and source of this FLP recombinase is 2 micron circle plasmid of DNA of yeast. Okay, so this is the CRE logs or FLP FRT system, which is a site specific recombinase and less complex. Okay, so tyrosine recombinases are involving the CRE recombinase, FLP recombinase, and the integrases class has this ATTP, ATTB sites in it, recognition target. Okay, next go to our question. So, to which family of enzymes do CRE and FLP belong to? So, we just now saw the table. There are 4 families, and CRE and FLP belongs to A1 tyrosine recombinases. Okay, so the correct option will be tyrosine recombinases. Next, we have the Phage DNA, contains an attachment sequence called, and the bacterial DNA containing the attachment sequence called. Okay. So we just now discussed what was with the attachment sequence called in case of phage DNA. It will be, yeah, for phage it will be ATTP, so ATTP, and for bacteria it will be ATTP. This will be the correct option. Okay, so the FLP is a site-specific recombinase encoded by dash circle of yeast Saccharomyces cerevisiae. So it is a 2 micron circle. 2 micron circle. It is encoded by the 2 micron circle of the yeast Saccharomyces recombinase. Okay. Yeah. So these are the all questions that I had today. Anything else you want to ask me? Anybody has any question? Was it clear to everybody today? Yes, ma'am, it was clear. Okay, good. Okay, so if you have any doubt, you can ask me, or in the next session also you can ask me. Okay? Yeah. And just one question. Hmm. Yeah, in the initial recognition step. Mm-hmm. Hmm. Yes. So here what, like it is like this, right? 5 prime, 3 prime. After the broken DNA, I'm sure. Okay. Okay. So now here at both the ends it binds, right? It stops. Okay. Yeah, so it in both the direction it is binding, right? So as it is moving towards this direction in this strand, and it this broken DNA, it is moving in that direction. So at both the direction, as I told, this particular strand here, if we'll see, as it moves, this one it cleaves more vigorously. It has some preferences like that, these protein, and this one is being fragmented less. And when the chi recognition site reaches, then the protein's ability to cleave this particular portion, the lower strand, which will create a deep 3' overhang, stops, and only the upper portion, which will generate the 5' overhang, will remain. Okay, so that is how in both the direction it will perform the similar activity. Here also it will cleave only 5' and generate the 3' overhang. Similarly here also it will create a 3' overhang and cleave the 5' end of the DN. I hope it clears your doubt. Hmm. Mm-hmm. For 5, yeah. Yeah, actually this protein is initially a bipolar protein. Okay, in the initial, but once the recognition site, the chi site is recognized, the polarity is lost, then it only cleaves only in one direction. The other protein or the other direction is that ability stops. That is why the 3' overhang is generated. Mm-hmm. Hmm. Hmm. Yeah, yeah, yeah. Okay, good. Anybody else? Okay, if there's no doubt, we'll conclude today's session. Okay. Okay, then thank you for joining. See you all next week. Bye.